Hello, welcome once again to our channel Bioholic with your Friday educator Olivia. After the first part of locomotion and movement, today I am going to start the second part, and in this part I will describe you what is skeletal muscle and about its structure. So let's start. In the last part we have learned about the three types of muscles. Those are the visceral muscle, cardiac muscle and the skeletal muscle. Let's compare these three types of muscle before starting to learn about the structure of skeletal muscle. The skeletal muscle is associated with the bones, but the cardiac muscle is present in the heart and the visceral muscle is present in the visceral organs. The shape of the skeletal muscle is cylindrical and it is unbranched, but the cardiac muscle is branched. And the visceral muscle is also unbranched, but they are spindle-shaped. The speed of contraction of skeletal muscle is fast, but the speed of contraction in cardiac muscle it is intermediate. And in case of visceral muscle, the speed of contraction is slow. The skeletal muscle is a voluntary type of muscle, but the cardiac and visceral both are involuntary in nature. And in both skeletal and cardiac muscle, cross striations are present, but in case of visceral muscle, there is no cross striations. That's why it is also called as smooth muscle. So as a summary, we can say that skeletal muscle is found attached to the skeleton and is responsible for the voluntary movement of bones, and skeletal muscle fibers run in parallel tracts and are multinucleated and heavily striated. And most skeletal muscles are attached to bone by bundles of collagen fibers known as tendons. This is a typical structure of skeletal muscle. We will learn about every part of this in details in our next slides. Now at first let's learn what is muscle mass. Muscle mass or muscle tissue is made up of a large number of individual muscle cells or myocytes. Muscle mass is separated from the neighboring tissues by a thick fibrous tissue layer known as fascia. And in the muscle fibers are arranged in various groups called bundles or fasciculi. Beneath the fascia, muscle is covered by a connective tissue sheath called epimysium. Connective tissue sheath that covers each fasciculus is called as perimysium, and each muscle fiber is covered by a connective tissue layer that is called as endomysium. And what is muscle fiber? The muscle cells are commonly called as muscle fibers because these cells are long and slender in appearance, and each muscle cell or muscle fiber is cylindrical in shape. Average length of the muscle fiber varies between 1 cm to 4 cm depending upon the length of the muscle, and the diameter of the muscle fiber varies from 10 micron to 100 micron. And muscle fibers are attached to a tough cord of connective tissue that is called as tendon, and tendon is in turn attached to the bone. So here you can see this is tendon, this is epimysium, this is endomysium, here is the fascicle, here is perimysium, and by the tendon the muscle is attached to the bone. Clear? Now each muscle fiber is enclosed by a cell membrane called as plasma membrane that lies beneath the endomysium, and it is also called as sarcolemma, and cytoplasm of the muscle is known as sarcoplasm. The sarcoplasm consists of nuclei, myofibril, Golgi apparatus, mitochondria, sarcoplasmic reticulum, ribosomes, glycogen droplets and lipid droplets. Now what is myofibrils? Myofibrils are the fine parallel filaments present in sarcoplasm of the muscle cells. In the cross section of the muscle fiber, the myofibrils appear like small distinct dots within the sarcoplasm. Diameter of the myofibril is 0.2 to 2 micron, and the length of the myofibril varies between 1 cm and 4 cm. Now let's zoom into the microscopic structure of the myofibril. Light microscopic studies shows that Each myofibril consists of two alternating bands, light band or I band and dark band or A band. I band is divided into two portions by means of a narrow and dark line called Z line or Z disc, and the portion of myofibril is between two Z lines is called as sarcomere. In the middle of the A band there is a light area called H zone. In the middle of the H zone lies the middle part of myosin filament. This is called as M line. In this picture you can see the I band and A band. In the I band you can see the Z line or Z disc. In the middle of the A band there is the H zone, and in the A zone here in the middle there is the M line, and the portion of myofibril in between two Z lines is called as sarcomere. Again here I have given the light microscopic diagram of the same. Now the differences between A band and I band. The A band is also called as anisotropic band and the I band is called as isotropic band. The A band appears as dark band under the microscope and the I band appears as light band under the microscope. The A band has H zone, that is a wide light zone, but the I band has Z line, that is a cross made by a membrane. In case of A band, length remains unchanged during muscle contraction, but in case of I band during muscle contraction, it is shortened. Now the myofilaments. Sarcomere consists of many thread-like structures that is called as myofilaments. Myofilaments are of two types, the actin filament and the myosin filament. Both actin and myosin function by controlling the voluntary muscular movements within the body, along with the regulatory proteins known as troponin, tropomyosin and meromyosin. They are also responsible for both cellular movements and non-cellular movements. Some lateral processes called as cross bridges arise from each myosin filament. These bridges have enlarged structures called myosin heads at their tips. Myosin heads attach themselves to actin filaments, and these heads pull the actin filaments during the contraction process. Here you can see the cross bridges and the myosin head in its tips that is attached to actin filaments. Now the contractile proteins of muscles, specifically of skeletal muscles. Myosin filaments are formed by myosin molecules and actin filaments are formed by three types of proteins called actin, tropomyosin and troponin. These four proteins together constitute the contractile proteins or the contractile elements of the muscle. In addition to the contractile proteins, the sarcomere contains several other proteins that play various important functions. They are actinin, desmin, nebulin, titin, etc. And at last, another important part of the skeletal muscle is the sarco-tubular system. The sarco-tubular system is a system of membranous structures in the form of vesicles and tubules in the sarcoplasm of the muscle fiber. It surrounds the myofibrils embedded in the sarcoplasm. Sarco-tubular system is formed mainly by two types of structures, the T-tubules and L-tubules or sarcoplasmic reticulum. The T-tubules are transverse tubules or the narrow tubules that are formed by the invagination of the sarcolemma, and the L-tubules are the longitudinal tubules, are the closed tubules that run in long axis of the muscle fiber forming sarcoplasmic reticulum. They do not open to the exterior like T-tubules. And what are the functions? The T-tubules are responsible for rapid transmission of impulses in the form of action potential from sarcolemma to the myofibrils. And the L-tubules store a large quantity of calcium ions. When action potential reaches the system of L-tubule, the calcium ions are released into the sarcoplasm. Thus, the T-tubules and L-tubules both help to trigger the processes of contraction of the muscle. So that's all about the structure of the skeletal muscle. If you have any doubts or any queries, you can ask me in the comment section, or you can also mail us directly to our mail ID that is provided in the description box. The self-assessment test link of today's class is also given in the description box. Check it and don't forget to test yourself. So thanks for watching. See you in the next class. Till then, stay tuned with Bioholic.